I'm Dr. T.R. Ganesh Babu, Professor ECE, Muthiammal Engineering College. Today, I will cover about retinal image processing. First of all, I will discuss about detection of glaucoma using fundus image and a optical coherence tomography image. This is my focus of work. Introduction: detection of glaucoma using retinal fundus image, detection of glaucoma using OCT image, detection of glaucoma with combined features. Finally, I will conclusion and future works. First of all, we are discussing about need for study. Glaucoma does not show any symptoms. First is affect the side vision, then total vision will be affected gradually. And of all, this type of research work to reduce the stress and time management of the ophthalmologist. This is the overview of glaucoma. Glaucoma is a serious eye disease which causes blindness in the worldwide. A watery material is called aqueous humor present in the eye. Aqueous humor drying out, the pressure will be produced in the eye. Due to pressure, a nerve to be damaged. This is called glaucoma. In first image is a normal one, no drainage, any fluid, so no glaucoma. In second one, pressure will be increases, drainage will be occurs, therefore the fluid will be drained out, so pressure will be produced in the eye. Due to the pressure, nerve to be damaged. This is called a glaucoma. This is examination of the glaucoma. First of all, ophthalmoscopy lens. Next one is tonometry, visible field test. Next one is imaging technique. In imaging technique is consists of two types. One is called fundus image, another one is called OCT image. First, in ophthalmoscopy lens, that is a lens. Doctor look out to the color of the nerve. Based upon the color of the nerve, it will be decided it's maybe it's glaucoma or not. But it is only for subjective analysis. So we are moving to another test, tonometry. Tonometry is to measure the interocular pressure in the eye. In normal range of the pressure is 10 to 80 mercury level. More than 80 mercury level, it may be is glaucoma, so nerve may be damaged. But it is two type of glaucoma, is the hypertension glaucoma and a normal tension glaucoma. In normal tension glaucoma is pressure is normal, but nerve to be damaged. That is called normal tension glaucoma. In hypertension glaucoma, the pressure is higher, but nerve is not to be damaged. That is called hypertension glaucoma. Therefore, this test is not 100% calculation of the results. Then we are moving to visual field test. In this test, it is useful to analyze the side vision of the eye. First, a moving light passed across the patients. The patient, if see the light, it will be press the switch. Then computer to draw the map. Based upon the map, only what angle the power will be lost, it will be decided. Based upon all the test, doctor is maybe conclude whether it is glaucoma is there or not. But all the tests also invasive test. So now we are moving to non-invasive test. It is called imaging technique. In primarily, two type of imaging is available in the retinal image processing. One is called fundus image. Another one is called optical coherence tomography images. This is fundus image. The outer portion is called a optic disc, and the inner side, the bright portion, is called optic cup. This is the blood vessels and the optic nerve. In all the nerve you converge at the point in a disc, one point in a disc. Due to the pressure, the nerve damage, yeah, some bright damage, some hole will be formed inside of the optic disc. It is called optic cup. This portion is the brightest portion. This is another one of the image is called optical coherence tomography image. It is a cross-sectional image. In this image, the outermost boundary is called retinal filter boundary. That is in inner side. This is called retinal choroid boundary. First, identify the two boundary. In the two edges of the choroid distance is called optic disc. A line joined from the choroid, that line intersect with the RB boundary in left side and right side. In between these two distance is called optic cup. So then we are measure cup to disc ratio. Actually, cup to disc ratio is one of the parameter to measure the glaucoma. It is a 0.1 to 0.3 is a normal patient. In more than 0.3 is called glaucoma. But in some patient having large number of disc size is very large, but value is very less. That is in between 0.1 to 0.3 is maybe a glaucoma. In some patient, disc will be small. The ratio greater than 0.3, it's not a glaucoma. Therefore, this parameter is not only for consider the examination of the glaucoma. Then only we are moving to OCT image. So this is my objective of this my work. First, to determine the cup to disc ratio, ISNT, inferior, superior, nostril temperature from the fundus image. To determine the cup to disc ratio and other features from OCT images, detection of glaucoma using all the combined features, that is fundus and OCT image features. In a first, we are discuss about glaucoma detection using fundus image. First, this is a first feature is CDR, optic disc. Next, optic disc. First, we are find out the two algorithm, hill climbing algorithm with the K-mean clustering, and the optic cup we are using three type of algorithm, that is called K-mean clustering, fuzzy C-mean clustering. The ISNT ratio for retinal blood vessels are optic disc, and ISNT ratio are determined retinal neurorim. These are the features now we are discuss. This is complete overall proposed block diagram of our system. First of all, we are segment the optic disc using fundus image. This is a input fundus image. First, define the image size, 1000 cross 1050. This image is RGB image. All raw image captured from the camera is called RGB. From the RGB image, to separate the three channel, R channel, G channel, and a B channel, This is the separate this three channel. In visually, we are look in this all the channel. Green channel is visible one compared to other channels. So in this channel, search the maximum bright spot in the image from the brightest part to make a rectangle 360 pixels from right, left, top to bottom, all side, because our region of interest is only in this portion only. So our input image size is 1000 cross 1050. Based on our input image, I will reduce the size in the size 360 cross 360 for time consumption purpose. K-mean clustering algorithm. It is one of the powerful clustering. It is a color clustering to say, identify the clusters based upon the intensity. Find the distance between the samples and the cluster means. Then assign the samples in the cluster with which the distance is minimum based upon the intensity. Calculate the new mean of the cluster after sample is added. Repeat the steps till the predefined threshold will be set, because you set the some threshold level. This is the input ROI image. First, I will assign the cluster center 3. 3 clusters will be detected. First one, second one white, and third one black. So I will trace the from this cluster, corner cluster will be removed. So remaining two cluster clubbed and trace the boundary, but a boundary is not obtained properly from this cluster. Suppose I will set the cluster is 4. So at that time, some extra regions also will be detected. It is a very difficult one, set the cluster value, because our blood vessels and the background also same intensity. Therefore, threshold level setting is very difficult to one. So to avoid this problem, automatically set the cluster we are using for another hill climbing algorithm. This is another one, k equal to 3. This is k equal to 4. Is a boundary. This a big boundary is called optic disk. Is an optic disk boundary. K equal to 4. To select the proper cluster value, so you automated the cluster value. So we are going to hill climbing algorithm. Next section, we will continue from hill climbing algorithm. Thank you.